Now ready to begin today's webinar on how to use the Haney Test. Our presenters today are Dr. Anna Cates, Extension Specialist, and Liz Stahl, Extension Crops Educator. Just one more note before we begin. If you need CEUs for participating in today's webinar, you'll need to view the entire webinar and complete the evaluation that you'll be redirected to when you log out of WebEx today. And even if you don't need CEUs, we ask that you complete the evaluation. We do use this information to improve future trainings like this one. Anna, I'm going to go ahead and turn that over to you now.
Okay, thank you, Jenny. As she said, I'm Anna Cates. I'm an Extension Soil Health Specialist as part of the University Minnesota Office for Soil Health. And my new office at the university dedicated to soil health education for farmers and conservation professionals. So we're really pleased to see the high level of interest for this program. My contact information is on this slide and at the end, and I welcome any comments you have about this webinar or soil health education needs in general. So soil health is defined by the NRCS as the capacity of soil to function as a living ecosystem. And really we know it when we see it. In these photos of soil that's been soaked in water, we can see the structure of both this near pile and this far pile is much higher than the one in the middle, which is really just a puddle. However, I would say those ones on the edge are healthier soils. But there's also a push to measure soil health, and there's a number of ways to do that. So I'm going to talk today about a suite of measurements known as the Haney Test after Dr. Rick Haney, who developed them. And this presentation is going to be fairly heavy on data, but there's a lot of data in there. This is a snapshot of one of my data spreadsheets, which has 30 different columns of measurements. I certainly won't be able to go over every one today. I'm going to focus on carbon and nitrogen pools, which is where my research interests lie, and then Liz will get into phosphorus and potassium as well. So our goals for today are to explain methods for the Haney measurements of carbon and nitrogen. I got a great pile of data from Test labs, thank you for that, where I'll be able to show an average range from 2017 and 18 Minnesota, mostly a few Iowa samples. That'll give you a sense of whether your data is sort of in that range. And then Liz will be discussing the use of Haney for fertility recommendations. So first, what are we measuring? I think of soil health tests as an attempt to measure what the microbe experiences. This is a picture of the food web underground. Plant residue is the primary source of food for everyone underground, but microbes can only use plant residue that's already partly broken down into smaller molecules. So that's this arrow here, where something has already digested the organic matter and excreted it into smaller pieces that microbes can enjoy. Microbes do feed directly on plant molecules, especially root exudates, which are simple carbon compounds that plants spit out from their roots while they're photosynthesizing. And then microbes also feed on their own waste products. They essentially recycle their biomass for the next generation. So in order to guess what microbes are up to, we have a few options. We can try to get a handle on how much of that simple organic matter is available for microbes, and that's both the carbon and the nitrogen there. We try to exclude more complete plant residue, more complex forms of organic matter, and just get the simplest stuff. The other way to track microbial activity is to look at their waste product, carbon dioxide, and that's what the CO2 burst test measures. So those are the two main pools I'll be talking about today. So Haney starts big picture with soil organic matter, and this measurement is pretty standardized throughout the soil testing world. It's done by heating soil so completely that any organic material is burned away as gases. And the mass loss after that heating is the mass of organic matter. It's usually expressed as a percent. And this histogram shows the number of samples in the MVTL data set at varying levels of organic matter. So there's about 200 here at this 2.5% level and about 600 here at this 5.5% level. This is relatively high compared to the rest of the country. This map shows darker areas with higher organic matter, and you can see Minnesota has some of the darkest colors on the map. That's because we combine high clay content, which holds organic matter, with lots of leftover organic matter from prairies and forests prior to agricultural lands. And then we have a long enough winter that decomposition happens relatively slowly. So percent organic matter is tied to a lot of things, but especially with soil texture, another really important property. Texture data samples. But since we wouldn't expect a 2% organic matter soil to behave like a 6% organic matter soil, for the rest of the data, I'm going to divide it into three groups. There's going to be high organic matter data, above 5.5%, medium, and then low organic matter data. So all the rest of my plots are going to look like this. I have these three panels, one on the left for the low organic matter data, in the middle for the medium, and on the right for the high. And then in this case, I'm looking at water extractable carbon, but that'll vary from slide to slide. So these box plots work by showing you the median as this line in the middle, and then the bottom of the box plot is the 25th percentile, and the top of the box plot is the 75th percentile. So 95% of the data is encompassed here in these lines, and any dots like this are statistically determined to be outliers. So you consider the really average range to be within the box and the quote normal, but encompassing the extremes to be everything touched by a line. So this pool of organic matter is water extractable carbon, which is similar to what it sounds like. You simply shake soil with water for a designated period of time. There's not a report for how long for this particular lab. And then you measure the dissolved carbon in the water. That's what this funnel is showing. This soil has been shaken, and then whatever carbon comes out in this water extracted is the ppm represented here. This makes sense as being relevant to microbes because microbes live in water films, and so the nutrients they can access are mostly in water. Keep in mind that this is only a small part of the total carbon pool because a lot of carbon will remain bound to minerals and not be measured by this process. Some other studies I looked at found that this was less than 1% of total carbon or less than 5% of the total carbon. Ward reports, Ward Labs does a lot of the testing for Minnesota Valley Testing Labs, so I'll be referring to Ward Labs testing procedures too. And they report that most of their samples fall between 1 and 300 ppm for this measurement, and Minnesota is right in normal for that. The next assay I'm going to talk about is soil respiration, which is sometimes called a CO2 burst test. Solvita works on this same principle, and the way you do it is you just put a cup of soil in a jar. That's this little plastic cup inside a mason jar in the picture. You moisten it to a moderate level, and then you seal the jar. The sealing is really important. You keep it sealed for a designated amount of time, usually 1 day, as in here. And then you measure the concentration of carbon in the jar. That's all the CO2 that the microbes have breathed out as they decompose organic matter. So sometimes you'll see this reported as parts per million per day as a rate, and in this case, because it's just 1 day, they just report parts per million. The Solvita paddle is designed to change color at different CO2 concentrations, but in my case, in this picture, we use an infrared gas analyzer, which I believe is what Ward does. But keep in mind that this isn't an absolute rate of decomposition by microbes. They're kept at a constant temperature and moisture for a short period of time. Sometimes this measurement is called potentially mineralizable carbon, which I think is nice and explanatory because it represents the potential of the soil microbes to decompose carbon under ideal conditions. So how do we look here in Minnesota? This one is really correlated to organic matter, which is true in all research study. There's really long upper limits to this data, so lots of outliers up high. And that's probably because you had a sample that has a big chunk of fresh residue or manure or an invertebrate carcass, and that would cause a brief or intense flush of CO2. Ward Labs suggests 200 ppm as a cutoff for very high samples, so I'm going to focus on the values below that. So here I have all the data, and then the next slide just shows only the data under 200 ppm. And here are the categories listed by Ward Labs from below average to above average to high. And Minnesota looks really good in regards to soil respiration, which I think goes back to our looking really good in total organic matter numbers. Average is right here at 50 ppm, and even the low organic matter samples are mostly in the average or slightly below average realm, whereas the medium and the high organic matter samples are mostly above average in terms of soil respiration, according to the Ward Labs categories. So this measurement kind of embodies both microbial activity and food source, but I think what we're seeing here is that it's so well correlated with total food source or total organic matter. That's really what this measurement is getting at. Now we get into slicing and dicing the measurements a little bit, and I think this is where the Haney test gets more confusing and starts to proliferate its columns. So sticking with carbon, this is what they call microbially active carbon. It's the product of dividing total soil respiration, which is the number we just saw in ppm, divided by water extractable carbon, which is the number before that. So this is assuming that water extractable carbon represents the total potential food source, and the soil respiration is the quantity that microbes could access. So how much of that total food source is microbially active? But it goes over 100%, you see, even in low organic matter soils, because physically bound carbon may also be active or accessible to microbes in that 24-hour respiration. And then once again, that 24-hour respiration doesn't represent total microbial activity, it's just a potential, more or less. So our soils here fall kind of between 25 and 75% microbially active carbon. And Haney's guide suggests that 50 to 75% is ideal. And that range means that there's sufficient carbon in that water extracted carbon, and microbes are able to use it. But they're not using all of it, so there will be some in the future. The idea is that if you go too high in this percentage, your microbes are going to be working through that water extractable carbon too rapidly. But that ignores the fact that these pools can constantly be replenished by surface residues and living roots in the ground. And that's why we harp so much on living roots in the ground in a soil health system, because plant residue is the source of all these pools of carbon, and those living roots are the best source for microbes. Next, we get into nitrogen. Water extractable nitrogen is measured in the same procedure as water extractable carbon. Again, we have the funnel, the shaken water soil slurry, and the nitrogen in the water that slips out. However, they present organic nitrogen as that total water extractable nitrogen, and then they subtract nitrate and ammonium nitrogen, or the inorganic pool. And in the Haney labs, or when labs are following the Haney procedure, they extract soils with extract nitrate and ammonium with weak acid instead of KCl, so the numbers are not going to be exactly the same as they are in your standard soil test. So we expect that this water extractable pool is going to be larger than just nitrate because it contains proteins and other organics that are a nitrogen source to microbes. Keep in mind that this organic nitrogen can also be taken up by plants, although that's less common and more difficult for plants. Then they have this organic to inorganic nitrogen ratio. So it's the number we just saw, which was 15 to 20 ppm, divided by nitrate ppm, which can be around 15 to 20, or it also, in this data set, goes as high as 300 ppm. And the idea is that you need some of both, because inorganic nitrogen is easier for plants and microbes to use, but organic nitrogen is the source of that inorganic pool. The Haney test suggests that above 5 is something to aim for, and that's a long way to go for most of these Minnesota soils. Above 5 would indicate that the vast majority of nitrogen circulating is not in the nitrate or ammonium forms. So that's something you would see, especially if you weren't applying any inorganic nitrogen fertilizer, whereas below this ratio, meaning you have more inorganic nitrogen, would indicate that there's more nitrate and ammonium than organic nitrogen. I would expect that if you just applied inorganic nitrogen fertilizer. So our soils are looking fine here. One interesting thing is that all of the different levels of organic matter have a similar ratio of organic to inorganic nitrogen, which I think means this has more to do with fertilizer dynamics than the organic matter dynamics. The next ratio is maybe a little bit more of a familiar concept in the soil health world. This is the carbon to nitrogen, which we usually use in terms of residues. But this is the C to N ratio just of that residue or of that soil water extract. And just like with residue carbon and nitrogen, C to N close to 10 to 1, close to that microbial biomass CO2 is ideal. That's what's going to promote microbial activity without causing them to mine the soil for nitrogen. Ward Lab suggests that they worry about ratios higher than 20 to 1, which means that the soluble organic matter isn't accessible to microbes without them looking for more nitrogen. And they suggest that anywhere 8 to 15 is good. So most of our Minnesota samples fall in that range. And once again, keep in mind that while residue C to N levels tend to be much higher because we're talking about full plant biomass, This is something that's already been pre-processed or leached from that residue. It's already soluble. So it's going to look a lot lower, and that's okay. That's good. One of the trickier calculations in the Haney test is the nitrogen release. There are some old versions of this formula out there, which makes it even more difficult to track what's going on with this calculation. But I did confirm with Ward Labs that this is what they're using. They have water extractable nitrogen times that percent of microbial active carbon times 4. So remember, microbial active carbon is the proportion of soil respiration burned off, or the proportion of water extractable carbon burned off by the soil respiration test. 4 is the average number of times in a growing season that nitrogen will be released in a significant rainfall. So there's assumptions built into this. One is that microbes will use a similar proportion of water extractable carbon as they will nitrogen, which is going to be true if your C to N ratio is right in this ideal area. Otherwise, your microbes are likely going to use a lot more carbon than they will nitrogen. The other is that number 4. Again, that average number of times in a season you will get nitrogen released from your soil. I haven't done any research on this topic, but I think all of you who experienced this 2019 growing season understand that some years you will have many more rains to release nitrogen in your soil than 4. So that's one thing. Another important thing is that the total nitrogen release can't exceed the water extractable organic nitrogen. So if this calculation kicks out a number higher than that original water extractable organic nitrogen number, it just is knocked back down to water extractable nitrogen. So one thing I was interested in is if you're going to slice and dice the numbers this way and look at these calculations, I was wondering how the calculated nitrogen release might change if the components change. So I did some example calculations with just the medium organic matter data to show how this might work. So in this case, the first box is the standard nitrogen release, around 25. That's exactly this number. Then I doubled the water extractable organic nitrogen and recalculated it, and you see a little bump in the predicted organic nitrogen release. Then I doubled the microbial active carbon percent, and you again see a little bump, but not quite as large of one. So there's the largest effect of doubling water extractable organic nitrogen because that represents the limit for nitrogen release. Microbial active carbon tends to be a little bit more variable because it's related to soil respiration, which I'll talk about more later. But I think if you see a number you don't expect in this predicted organic nitrogen release column, I would go back to your water extractable organic nitrogen number, see if that's especially high or low, and see if you've hit your limit essentially, and that your nitrogen release calculation is the same as your water extractable organic nitrogen. The next calculation I'm going to the soil health calculation. This is how the soil health score is currently calculated using respiration, water extractable carbon, and water extractable nitrogen. There have been other versions that also use that C to N ratio, but since 2017, this has been the formula. And the suggested interpretation is that over 7 represents a good soil health score. And most Minnesota samples fall easily above that range, which brings home the importance your field sample to one that you take on the same soil type but with improved management or in a natural area like a fence row. This gives you something to realistically compare your agricultural soil to, more so than this national average of 7. So there's no clear reason why you would divide these values by 10 or by 50, except just to emphasize their relative weight. A little contrary. I did the same exploration the soil health score to get a sense of which of the measurements used in the calculation had the most effect on the overall score. So once again here, the first box represents the standard score from the medium organic matter soil, so it's exactly this number. And then in the next box, I doubled the nitrogen value and the water extractable nitrogen. In the next box, I doubled the water extractable carbon. And in the last box, I doubled the soil respiration number. And importantly, you see this increase when you double soil respiration, but you also see the overall box get bigger, which means the range of numbers is higher. You can see here that the range of values in soil respiration is higher. It's about 100 compared to a 50-part range for water carbon and a 15-part range for water nitrogen. I'm going to talk a little bit now about sources of variability for soil respiration. First, sample processing. This part isn't going to be your responsibility in the field, but studies have shown that different sizes of sieves and different methods of re-wetting dried soil have an effect on soil respiration numbers in the lab. So it's inherently going to be variable depending on your processing. That means if you want to compare results across years, make sure you stick with the same lab and make sure they're sticking with the same method. It's perfectly reasonable to call and ask. The other thing is that some soils are inherently more variable than others. There was a the variability of this measurement specifically that showed that. And it has to do partly with soil structure, shown here as aggregates in different size classes. Aggregates are destroyed by sample processing, releasing carbon in a big flush. So if you have a soil that's really well aggregated, it's going to show that flush more clearly. The other part, which is going to be the responsibility of the sampler in the field, is topography. These are soils from our Life in the Pits field day. One is upslope or mid-slope, and one is more downslope. And you can see that there's a shallow but rich A horizon in that upslope position and a much longer A horizon in this eroded, depositional area where everything had eroded into this area. And you can imagine just from the color that the top 6 inches of these soils would test differently. You'll see it in the field that the plants look different. So when you're taking your samples, make sure to be consistent in where you collect from. This is sample timing. This is from a set of samples I took at the Lamberton Southwest Research and Outreach Center this summer. I sampled twice a week and I got similar average values for soil respiration every time, but some days were much more variable than others. And this has to do with the environment the microbes were experiencing prior to sampling. So for example, these days were pretty variable, and then this day and this day were a little less variable. And we are still trying to figure out how to constrain that variability. By that is how to make sure we sample in a way that's going to allow us to see differences between different management systems or sites. Based on this research, I'm curious when those of you who have used the Haney test have sampled during the year. So this is another quick poll question. I'm curious whether you sampled A, early in the spring, B, at a pre-sidrous nitrate test sample, so essentially when the corn is already up, later in the spring. C, early, or D, fall but close to frozen, so last thing in the year, or E, you have not used the Haney test. There's no right answer. Your answer will not be used against you. Don't worry about that. Okay, so it looks like a lot of people are hitting that late spring window, slightly more in the late spring or spring than in the fall. Once again, there is no right answer. The most important thing is that you try to match conditions each time you sample so that you're comparing apples to apples, and you're not trying to say that has drastically improved because you sampled 3 weeks later and it was warmer and the soil respiration was much higher later in the year. Okay, I'm going to hand off to Liz here pretty soon, but I'm going to leave you with this image of the soil organic matter food web again. I'll remind you that we didn't come anywhere close to hitting all 30 columns of that spreadsheet. And again, all the columns in that spreadsheet probably don't represent all the activity from the different soil organisms, not even my cartoon soil organisms. I hope you have a better sense of what the organic matter tests are trying to estimate, and also what the Minnesota averages are. That will be on the archive of the webinar. And then what kind of variability to expect and why. Liz is going to talk more about using this data in a practical sense and giving some fertility recommendations, so a little different take on it.
All right, well, thanks, Anna. Well, again, I'm Liz Stahl, Extension Educator in Crops with the University of Minnesota. I work out of the Worthington Regional Extension Office here in southwestern Minnesota, and I'm going to talk today about a project we've done looking at whether or not soil health tests should be used in generating fertilizer guidelines. But first of all, I just wanted to talk a little bit about what got me interested in looking at soil health tests in the first place. When I started with Extension over 15 years ago, I got involved in a long-term tillage trial Well, we looked at strip tillage, no tillage, ridge tillage, and chisel plow, and a corn soybean rotation. One thing that really stood out to me was how striking the difference would be among the tillage systems, like how water would be standing in the chisel plow system, for example, where there was no standing water, and like the no-till and strip tillage plots after we'd have a rain. I'd have to go out and take stand counts a couple of times, and it was pretty wet in the plots. Well, I just about lost my boots literally in the chisel plow plots, but I could walk right across the strip till and no tillage plots with no problem. So I call this my, muddy boot test. Well, there were stark differences in infiltration that you could see. I could see these differences, but then again, how could we measure these? What was going on there? So that's what really got me interested in looking at soil health tests and how we could measure differences that we could see between these different systems. There are several different soil test options available today. Some of you might have heard about, of course, the Cornell, the Comprehensive Assessment of Soil Health, or the CASH system. Of course, the Haney Test, which we're talking about today. The Soil Health Institute, they also have developed what we call Tier 1 soil health measures. There are also other tests that I'm sure you might have seen, like the Soil Your Undies. That's a pretty great visual way to demonstrate differences between management practices. And then there's a tea bag method, for example, too. That's one that a couple of our extension educators here at the University of Minnesota, Shane Bujega and Claire Lacan, they're also looking at that. But again, we decided to focus on the Haney test since this is a test that we heard most people were using, and many farmers that were involved in NRCS programs are also using this test, and we're also getting a lot of questions about it. So this is what we used when we wanted to take a closer look at soil health tests and see how we could use those results. So one thing you notice right away, if anybody's gotten a soil health test back from the lab, and this is what I noticed right away. Now, not all the tests or the labs that do this in Minnesota put this, but the ones that we sent back from Ward Lab, we were getting results that included estimations of nitrogen savings by using the Haney test. Also, if you look in the Solvita website, it states right on the website that you should be able to reduce, fertilizer by 5 to 25% while still maximizing crop yield. So of course it's being implied that by using the Haney test, you should have some nitrogen fertilizer cost savings. I was also hearing from people that they wanted to use the Haney test so that they could use that to help fine-tune their nitrogen guidelines. So of course that really piqued my interest, being a crops educator, because I know that changing N application rates, that can really have a dramatic impact, for example, on corn yield and ultimately your bottom line. So when I looked at these numbers from my sample results, again looking at the calculated sample savings, I figured out they were assuming that you would use the rate of 1.2 pounds of nitrogen to produce a bushel of corn for your corn grain goal or yield goal. Well, of course, we haven't used yield goal in nitrogen guidelines for years in Minnesota, and I've been with Extension for over 15 years. So again, that raised a red flag as this is really not a valid comparison for Minnesota. But anyway, let's get back to, before I talk about the results from our research, I think it's important just to talk about a few basics about soil testing, just so we're all on the same page. And first of all, the first thing we need to remember is that a soil test, it's a relative measurement of nutrient sufficiency for plants. Also, soil test, it measures the amount of a nutrient that's proportional to the amount that is actually available for plants. And a key point is also that with different tests, different extractants are used to measure the amount of a nutrient in a soil sample. The Haney, like Anna was talking about, it uses water, a weak acid, the H3A extractant. Standard soil tests, they use other extractants based on the nutrient. And it's important to note that the amount that's measured by a soil test, the amount that's measured of a nutrient, it really has no meaning by itself. Soil testing procedures are standardized in what extractants are used. So you run a soil sample using this method, then you get a number on a sheet. Well, what does that number really mean? I could use orange juice, for example, run an extraction, see what I get out of the soil sample, I'd get a number, but what does that number mean? How can I use that? That's where correlation and calibration come in. They give those results meaning. So the first thing looking at, let's look at correlation. By definition, a test is considered correlated when lower yield and plant growth can be predicted at lower soil test values, and higher yield and plant growth can be predicted at higher soil test values. So we're going to look here. I've got an example from phosphorus. On the x-axis of the graph here, you see that we have results from the Bray phosphorus test, and on the y-axis we have percent of maximum yield. So this graph shows that how soil test values correlate to yield for soybean, corn, and wheat. And we have a separate line for each crop. You can see as the extractable phosphorus goes up, yield goes up to a point, and then yield will flatten out at the higher soil test levels. So we're not seeing an increase in yield with higher testing levels. So that dotted line represents the point where additional fertilizer would not economically increase yield. And then the next step of this is, or the key piece, is calibration. So that's a process of determining how much fertilizer is needed to meet nutrient needs of the crop at different soil test levels. So here's an example looking at nitrogen for a nitrogen calibration. Now, calibration trials are commonly conducted by universities. They're usually small plot replicated trials where different levels of an application of a nutrient, they're tested, and then researchers see what they get for yield at these different levels. And they typically include a stand or two of no application of that nutrient. Now, literally hundreds of these trials have been conducted at many locations over many years, representing many different environments. And the trials continue today, especially since we're seeing changes in our environment like increasing precipitation, which of course that can impact nutrient needs of a crop. Now the fertilizer guidelines generated from this work can be expected to vary by state. Each state, for example, has different soil types, environments, precipitation, parent material of soil, length of growing season, and so on. So the guidelines are not a one-size-fits-all. But it is important to note that there's little to no correlation or calibration work has been done with the Haney test in Minnesota and really throughout the Midwest yet. So again, getting back to the Haney test project here, we started this back in 2016. We ran it for 3 years. I'll mention right away that this project would not have been possible without the help of Dorian Gatchell with Minnesota Agricultural Services. He was really key in finding the sites and collecting the samples. Also, we had support from AgVise and the Minnesota Ag Fertilizer and Research Education Fund, or AFREC, for the first year of this trial. So again, this project was run on pretty much a shoestring budget. So again, thanks to the support of these peoples and groups and the farmers also that let us sample their sites over the years. Now, they were on-farm trials, again, conducted at 12 different sites over 3 years. We selected some sites that were very similar in soil type, but then we also selected sites that differed in productivity. We just wanted to see how Haney test results correlated to standard soil test results and if we saw differences over time. This was also a pilot project just to see if further work was warranted, if we could get the funding to do so. What we did is select a location in each field, georeference that location so we could go back there each year for 3 years. Took about 12 to 15 cores around that selected spot, taking a sample at 0 to 6 inches, and then we also collected soil from 6 to 24 because we needed that for the standard test to test for nitrate nitrogen. Now that 0 to 6 sample, we mixed that up, split it in half. One half went to be analyzed by the Haney test, the other half for the standard test. Samples were all sent off to the same lab for analysis. And it's important to note too, people have questioned about this. By definition, the Haney test, that is to be sampled at 0 to 6 inches. For standard tests for like phosphorus, potassium, organic matter, and other nutrients, they're also ran at 0 to 6. But if you also want to sample for residual nitrogen in Minnesota, it's recommended, you're supposed to collect a sample from either 0 to 24 inches, or you could do that 0 to 6, and then also collect a 6 to 24 inch sample. And that's again because research has shown that a significant portion of the nitrate nitrogen in that 6 to 24 inches in the profile, a portion that is available for the crop, so we should account for that. Now, a few other key things here, looking at differences between the standard test and the Haney test. First of all, again, the extractants vary. We've mentioned already the Haney test uses water and that H3A, which is a weak acid. That's supposed to simulate more what the plant experiences in the soil. That's what they say with this test. And then so for nitrogen, the Haney tests are measuring water extractable nitrate nitrogen, ammonia nitrogen, and organic nitrogen, where the standard test, that's measuring nitrate nitrogen. And the standard soil test uses different extractants depending on the nutrient. So you can see that here off on the left-hand side. It's also important to note that for phosphorus, the Bray test is generally used if pH is less than 7.4, while the Olsen is generally used when the soil pH is greater than or equal to 7.4. This is not differentiated with the Haney test. Now, costs do vary by test, of course, too, and also if you can get discounts or not. But typically, the Haney test, that'll cost anywhere from $50 to $70 per sample. Whereas, for example, University of Minnesota, we charge $15 per sample for the standard basic analysis. If you want to run the test for nitrate nitrogen, and that includes that additional depth down to 24 inches again, then that cost will be another $8, so a total of $23 per sample. Private labs run about in that range too, give or take. It just kind of gives you an idea of the range and cost. And if you're just collecting the standard basic soil tests, the sampling depth would be the same between the Haney and the standard. But again, if you want to get nitrate nitrogen, you want to collect that sample down to 24 inches for the Haney test. Also sample handling, Anna mentioned this a little bit too, but it is a little more picky with the Haney test. You know, you don't want to, for example, leave the test laying on the dash of your vehicle, keep it cool and so on. So it can be a little more tricky with the Haney, but you do want to be careful with both. So we'll get into a little bit of the results here. And just for example, this is looking at the nitrogen first, comparing results of the Haney test to the standard testing procedures. So each dot that you're seeing on this graph, that is illustrating what we found at one location 1 year. So comparing again that nitrate nitrogen at that 0 to 24 inches versus what we found with the Haney test in that 0 to 6. Now the blue circles, that's representing results from 2016, the red diamonds, that's from 2017, and the green squares, that's from 2018. I tested the data to see how well results from the two tests correlated using a procedure called the Pearson's correlation coefficient. So basically, if the relationship was perfect, it would equal 1, or it could be -1. Here we're at 0.8758, so that's a pretty strong correlation actually. You know, 1 just really doesn't ever happen in real life, but that gives you an indication that, you know, these were pretty well correlated. But again, the next step would be, okay, how would it impact results if we try to use this for generating fertilizer recommendations? So what we did here, and I'm not going to get into all the details on this, but if you do want more information, you can go to our Extension Crops website. There's also different publications. For nitrogen, there's a nitrogen rate calculator that's housed at Iowa State. Basically with U of M nitrogen guidelines, again, we don't use yield goal. We calculate the maximum return to nitrogen. It uses a price ratio that's equal to the price of nitrogen per pound divided by the price of corn per bushel. That usually falls around 0.1, so I use that for simplicity. You can get rates for corn following corn or corn following soybean. Also, if you have sandy soils or irrigate, that can impact results again. And I also should note, you know, normally we wouldn't collect nitrate nitrogen samples unless you had situations where we'd expect you'd have more residual nitrate nitrogen. That would be, say, if you're in a drought condition or manure was used, but just an aside too. But again, I won't get into the big details of this. You can check out these websites for more information if you are interested in more details. So if we look at the 2016 data, for this comparison, I used a scenario where we were in a corn-on-corn situation. So this graph is showing the results of how much nitrogen would be recommended to be applied if we use the Haney test results, and that's in the maroon bars, versus the standard test, that's in the gold. And the comparisons are made for each of the 12 sites. We got the sites listed across here in the x-axis. Now in computing this, I took the target nitrogen rate that was calculated, that came out to 170 pounds of nitrogen per acre, then subtracted what was calculated to the amount of residual nitrogen in the soil that would actually be available for the crop. Now what we do for our standard testing is to take, you know, 60% of that and subtract that off our target rate. So when you look at our results, 8 of the 12 sites, we ended up that more nitrogen would be recommended if you use the Haney. And if we look at 2017, again, similar results in each case. 12 of the 12 sites, more nitrogen would have been recommended if you use Haney test results than our standard testing results. And 2018 here again came out to be 12 out of 12 sites. Some of these sites, like site number 8, we had a lot of big difference there. Had a lot of residual nitrogen, but again, the Haney test would have recommended having a lot more. Now, some people might say, okay, you're adjusting for this 60% of that residual nitrogen. With the Haney test, if that's supposed to be all available, let's not subtract off that 60%. So I did run that for each location as well, or each year, excuse me. And if you look at, I'm just going to show the 2018 for the sake of time here, we still end up with having more nitrogen being recommended if we use the Haney test to generate our guidelines versus standard tests at 9 of the 12 sites. It wasn't as dramatic, but still, again, it comes out to be more nitrogen with using the Haney test results. And some people might think that, well, maybe you should take a deeper soil sample than with using the Haney. But again, remember that's by definition only down to 6 inches. Whoops. And if we go ahead here, let's look at potassium, our results there. We did find a pretty strong correlation between the Haney test and the standard test. Our correlation's at 0.820 or 0.8202. So again, that's pretty strong. Now, if we take the next step and use that for generating fertilizer guidelines, here on the left we have the results from our standard test. Here's from the Haney test, and what happens with those numbers, we get put into different categories like very low, low, medium, high, or very high testing soils. I just put it into colors to help make the comparisons a little easier. And you can see how much fertilizer was recommended if we would use the standard test or the Haney test. And what you're seeing here is in all cases, we would have recommended a higher amount of potassium be applied if we would have used results from the Haney test. And I should note that for potassium, we actually do use yield goal in these guidelines. And I used a 210 bushel, which was pretty standard for this area. But again, we would end up having higher fertilizer application rates for all cases, and this was very standard across all the years of this trial. Excuse me. Now we look at phosphorus. This is a little different ball game here. We see our correlation coefficient is 0.529. That's a weak correlation. That's considered pretty weak. There's a lot of variability. You can see as we get higher testing levels in the standard phosphorus tests that we're getting a lot more variability in what we saw than with the Haney test. So again, not as very well correlated. This is where the phosphorus, we're going backwards. We're having low to medium, very high to high. This had flipped around very high to high. So again, you just see the range just not very well correlated. And again, that differences, sometimes we'd apply more fertilizer, sometimes we'd apply less with the Haney test. So not very well correlated. So just some key points here. So basically, again, the recommendation would be to use results from tests that have been correlated and calibrated in your own state. This way you can risk, otherwise you could risk having over or under application of nutrients. Also, whenever you get results back, ask yourself, okay, how are these recommendations that are listed on the soil test, how are they generated? Sometimes the lab will say you have university recommendations. Well, are they really from your state's university or somewhere else? Also, what are the rates that are being compared against if there's a quote-unquote savings? Again, what's the comparison? And I've heard this several times too, like on-farm trials, some people report good yields with less nitrogen. Well, what was their starting point? A lot of times when I ask questions further, they are actually applying more nitrogen anyway than what our guidelines would be. So if they cut down their nitrogen, by definition, they still should be getting optimal yields. So it's always good to ask, what's your starting point? So my wish list with soil health testing, I wanted to kind of wrap things up with that. My wish list would be, having soil health tests that are robust. You could use over multiple environments. They could work in many different soil types and conditions. The results would be repeatable. Won't matter so much, if you had a sample, cut it in half, you get very similar results with both halves, for example. Results would correlate to yield response. And you would be able to measure differences among long-term management systems. Again, we've tried this with different tillage systems, and unfortunately just were not able to detect big differences. Now, maybe we didn't have enough, we had corn on corn situation, but long-term tillage, maybe if we would have had, corn soybean rotation or cover crops, maybe we would have seen more differences, but we haven't yet in our standard agricultural systems. And also, I'd like something, of course, that's inexpensive and easy to use, have a sound basis in science in what's being measured and why, and also have sound correlation and calibration research behind them to give it clear direction and interpretation. So with that, we talked a little bit about all these different soil health tests. But one of the things, just getting back to what might be one of the best soil health tests out there, grabbing a shovel, dig up the soil, take a look at it. You can see physical differences like soil structure and things like earthworms. Shovel's not that expensive. You can use it over and over. Again, I think we are starting to scratch the surface in understanding soil health tests, and maybe, we'll be able to figure out in the future if we can modify our fertilizer recommendations based on soil health tests, or at least the health of our soil. But right now we're just really not there yet. So with that, do want to again thank all the people that were involved with this research and the project, and of course Bowser as well for hosting this webinar today. And with that, I'll open it up if anybody has any questions. Hopefully I didn't get you too confused by going backwards in that presentation there to the wrong graphs there between phosphorus and potassium. But any questions?
Hi Liz, this is Jenny. We did have one question come in related to your presentation. Can you talk about what were the management systems on the 12 trial sites? Were they mostly conventional systems or those that have applied soil health systems?
Yeah, and I've got to remember several of these, number of these did have cover crops with them as well. And I know that there were some differences, but they were in the CSP program, so that's why we were taking the Haney test anyway. So we just kind of wanted to find out more information about this. So yeah, some of these, the projects were, a lot of these already had cover crops with them.
Okay, thanks. And then Anna, we had a question come in earlier for you. Is the soil health equation used by Ward Labs or Haney or both?
So there are a few versions of soil health score calculations out there. Ward Labs switched their calculation in 2017, as far as I can tell, and then just in looking around at the other labs that use these suite of measurements, I think there are a few soil health scores out there. So depending on where you get your test taken, your interpretation of that score might vary a little bit, which is why I would encourage you to go back to those original measurements. What's the carbon? What's the nitrogen? What's the respiration? That's going to give you a better sense of what's actually going on in your soil rather than paying too much attention to the data after it's been sliced and diced various ways.
Okay, thank you. Um, we had another question come in. Does the soil health number have a benchmark goal to reach?
Yeah, so that's the interesting thing about running the soil health number for Minnesota soils is that they suggest that anything above 7 is good, and that represents, I think, 80% of the Minnesota data that I saw. Using that as a benchmark is probably a very helpful for most people. Rather, it's better to establish a benchmark by looking at a similar soil type. You can look at the Web Soil Survey to make sure you're on the similar soil type, or check with your SWCD or NRCS office, ask them to help you map that out. But using a benchmark from a similar soil type where someone has already improved management, or you're looking at a more perennial system like a fence row or a natural area, that'll kind of show you the maximum that your microbes might be up to on a similar soil texture, similar climate, similar landscape position, and all that stuff. And then Liz and my contact information is up there, and we welcome any feedback or questions that come up later. Thanks everybody for coming.
Yep, thanks everybody for listening in. And like Anna said, if you have any questions, feel free to drop us a email.